And we're very pleased to have Hannah Pizzato, PhD. She's coming to us from University of Arizona, from the lab of Deepta Bhattacharya in the Department of Immunobiology. And her talk today is going to be engineering human pluripotent stem cells to evade immunological rejection. Question of the hour. And a little bit about Hannah, a little bio. She received her BS in biochemistry from Purdue University. Then she went to Washington University in St. Louis for graduate school, where she joined Deepta Bhattacharya's laboratory. In 2021, she received her PhD in immunology, then continued to work with Deepta as a postdoc at University of Arizona. During her PhD, she worked on two projects, one examining the metabolic requirements of hematopoietic homeostasis, and another generating a human pluripotent stem cell line capable of evading immunological rejection. She has since expanded on both these projects during her postdoc to examine the role of metabolism in emergency myelopoiesis and to determine which immunological pathways require inhibition for human pluripotent stem cell persistence. Her work has led to first author publications for both projects, in addition to other co-authorships and 2 licensed patents. Congratulations, that's awesome. For engineering minimally immunogenic human pluripotent stem cells, and she hopes to continue working at the intersection of immunology and regenerative medicine in an industry setting in the future. Welcome, Hannah. Thanks so much for joining us. We really look forward to your talk.
Thanks, Monica, for the introduction as well as the invitation to speak here today. I normally do not speak to diabetes crowds, so feel free to stop me at any point if there is a little too much emphasis on the immunology side of things.
Yeah, well, I think I see several sort of like beta cell people and several immunology people, so I think there'll be some nice crossover.
Sounds like a good mix.
Great. Take it away.
To get started, I just wanted to go over the current barriers of transplantation, and many of you are probably already familiar with most of these. So one option for transplantation is to do an autologous transplant. In this case, cells are taken from the patient themselves and later put back in. And this is most commonly used for, hematological malignancies such as blood cancer, where healthy cells are taken out, the patient receives whatever chemotherapeutic treatment, and then the healthy cells are put back in to replenish bone marrow populations and mature cells from that. It can also be used for CAR T cell therapies, in which a patient's own T cells would be taken out, edited to become CAR T cells, and then put back into the same patient. As one can imagine, this is a very individualized process. You're taking cells from a single person and putting them back into the same person. It therefore doesn't scale up well, as the cells are patient-specific, and it can be incredibly costly, especially in cases, for example, like CAR T cells, where you're taking the cells out and individually editing them. The more traditional version of transplantation is using organ donors. In this case, you need a donor that is willing to donate their healthy tissues, and the patient is put on immunosuppressive therapy. As you can imagine, this also has issues with a limited number of people willing to donate organs, and some that are having healthy enough organs that they can actually be given to a new patient. Additionally, immunosuppressive therapies come with this whole slew of side effects, with a huge emphasis on an increased risk of infections as well as cancer. So in the current state of transplantation, the major barriers are how well we can scale it, as well as the issues associated with side effects from immunosuppression. And to get at the issue of scalability, a lot of people have turned their attention to pluripotent stem cells. For those who are not familiar, pluripotent stem cells are composed of induced pluripotent stem cells as well as human embryonic stem cells, so iPSCs or HESCs. What's exciting about pluripotent stem cells is they can continuously self-renew, and as they're self-renewing, they maintain their pluripotent state. In addition to this, however, many labs all over the world have figured out these really robust differentiation protocols, so you can differentiate pluripotent stem cells into multiple cell types, organs, tissue types, etc. So in this picture from a review, I'm just highlighting a few for those of interest. Many of you already know that we can differentiate stem cells into beta islets and other pancreatic tissues. But additionally, this goes far beyond pancreas, and you could also do, neuronal cells, different blood cell types, heart tissues, etc. So by using pluripotent stem cells, we go from a single dish of cells that we can differentiate, and that differentiation could be given to a ton of people. It's not this one-to-one issue that we have with organ donors or in autologous transplants. And as many of you probably know, stem cell-based therapies have already started entering the clinic. And this is probably pretty outdated at this point as the review is from 2020, but you can see that iPSCs and embryonic stem cells have both been differentiated into cell types to treat different diseases. For example, they've been differentiated into neuronal precursors that can treat Parkinson's disease, retinal pigment epithelial cells for macular degeneration, or as this audience is probably most familiar with, pancreatic cells, either the endoderm or beta islets, that can be used to treat type 1 diabetes, with the Vertex clinical trial probably being the most emphasized of the diabetes trials so far.
Agreed.
But the issue with this is that you still have immune rejection. So almost all of these clinical trials, the patients are still put on immunosuppressive therapies. So it's still great in that these patients can receive these tissues, but they have the nasty side effects of the immunosuppression. And the main drivers of immune rejection are T cells, natural killer or NK cells, phagocytes, and complement. And the immune system is a very complicated system of the body. So there are more cells involved than this, as well as more molecules, but these are the ones that drive immune rejection. Additionally, all of these cells can work together and work with other components of the immune system to drive this rejection. So the immunologists in the room are probably going to hate these very short descriptions of each cell type, but I wanted to provide it for those who are not immunologists. So T cells are the leading driver of graft rejection, and this is mostly driven by the fact that the graft from a donor is going to express the HLA present from that donor. And HLA is a highly polymorphic molecule, but the T cells in the patient are going to recognize this as foreign, and the T cells themselves will either kill those cells or they will prime other cells of the immune system to come in and attack. NK cells have this balancing act between activation and inhibition, and they receive signals from both. If the balance skews in favor of activation, the NK cells, as it suggests, will become activated and they will release cytotoxic granules towards the target cell. Phagocytes, which mostly consist of macrophages, again, as their name suggests, they phagocytose things. And this can include dying cells, dead cells, foreign cells, and so on. So they can literally come in and eat the cells out of a graft. And the final one I want to highlight is complement. And the complement cascade can be very overwhelming for some people, but the ultimate outcomes of the complement cascade is there are proteins that get released and they recruit in more inflammatory cells that can come and actually reject the tissue. And additionally, towards the end of complement cascade, you get the formation of a membrane attack complex, which pokes holes into cells and causes them to lyse. In addition to this, you also have antibodies, which themselves don't do a ton, but they act on other, or at least in the format of graft rejection, but they work on all of these other cell types, specifically NK cells, phagocytes, and complement to promote rejection and call in more immune cells to reject the graft. So our lab came up with an idea to generate a minimally immunogenic stem cell line. And I would also like to say that this project started over 10 years ago, even before I joined the lab. And during that time, other labs have also gotten in on this problem, and they've made their own strategies to address this same question. And, some of them have simply pushed the field forward a little bit, which is great. It's still progress, and we've included some of those edits in our cells, and others have even made successful cell lines as well. So this is a very exciting field that's making a ton of progress, and I'm sure in the coming years, we'll see even more and more ways that we can design cells to evade immune rejection. But today,
Can I just ask quickly, how broadly are these protocols shared across different laboratories? And is that a help or a hindrance in terms of pushing, the ultimate goal forward of generating this minimally immunogenic HESC?
So it's rather variable dependent on the lab. So, we've been pretty upfront when people ask us what our strategy has been. Some labs are as well, others are not, some of it's protected by IP. It is pretty helpful because as you'll see later in my talk, we have added some other proteins that we tested out. And some of that was based on other papers that came out or suggestions from people who don't necessarily work on this problem, but they work on one of these cell types. And they'll say, hey, here's a new inhibitor that's becoming or gaining more and more traction. So
Is there a certain repository for these types of protocols that people the go-to? I mean, obviously you can read in the paper, but some of the nuances aren't always shared.
Yeah, there's not a great repository that I know of.
Hmm.
Um, so we tried to have ours as apparent as possible in the paper, and we're also more than willing to answer questions if people contact us. And it really is based on who you know and how that lab handles their level of secrecy, if you will.
Yeah. Okay. Well, that's good. Good to know. Thank you.
Yeah, no problem. So what we did for our line is we started with a human embryonic stem cell line. We used CRISPR-Cas9 to ablate HLA class 1 and class 2, and this prevents the recognition of T cells, recognizing the mismatched class 1 and class 2. We additionally got rid of MICA and MICB, which is an activating ligand found on a lot of cells, especially in the pancreas. And this ligand activates NK cells that express NKG2D, which is almost all NK cells. By ablating MICA and MICB, we reduce cytotoxicity of NK cells by about 50%. In addition to this, we've added a slew of inhibitory proteins against NK cells to help seal that gap, as well as phagocytes and complement. And the end goal, because nobody necessarily cares to transplant stem cells themselves, is that they can be differentiated into other cell types, tissues, organs, and be used for transplantation without the need for immunosuppression. So to start off, I'm just going to talk about how we engineered our stem cells to knock out class 1, class 2 MICA and MICB. And so this entire workflow was done by transfecting our cells with Cas9 and various gRNAs. So the stem cell line of choice that we use are H1 stem cells, which are a highly characterized line of human embryonic stem cells. And the gRNAs that we use are beta-2M and TAP1, which would prevent the expression of HLA1, CD74 and C2TA, which would prevent the expression of class 2, as well as a guide RNA from MICA and MICB to target MICA and MICB, again for NK cell inhibition. So we transfect these into our H1 cells. We did this as an iterative process, otherwise we wouldn't have great efficiency if we tried to knock all of these out at once. After the transfection, we pick the colonies. Because this project was started a while ago, we did MySeq sequencing. There are better ways to do that now, but keep in mind, this has been a 10-year process. So we pick the colonies, we sequence them looking for indels in the targeted region by our gRNAs. And then just to confirm that we don't have mosaicism, we go ahead and do a single cell sort and then sequence them again. Throughout this talk, the cells that do not express class 1 and class 2 will be called HLA-1-2 knockouts. And those that do not express class 1 and class 2, as well as MICA and MICB, will be called HMKO for HLA-MICA-MICB knockout. And just to give you an example of the data we get from the MySeq, is that, it just gives you a sequencing section of the area of interest for the gRNA. And you can look for insertions and deletions, and you hope to have a frame shift mutation to knock the gene out of frame. And so here I'm just showing you an example of beta-2M, and you can see about 53% of our reads have a read of -1, which means a deletion of 1 base pair, and about 47% have a read of 5, which would be an insertion of 5 base pairs. So both of these would knock the gene out of frame. And by having about 50% representation of both, it suggests that both alleles of beta-2M have been knocked out of frame. And we can also test this by looking directly on if our cells express class 1 and class 2. So to look at class 1, we use a marker called HLA-ABC, which is just simply a class 1 molecule. As you can see from this plot, where we have an isotype control in black, our wild type cells in blue, and our knockouts in red. At steady state, there's not a ton of expression on our wild type cells, but you can still see that there is none on our knockout cells. To really push this to the limit, we treated our cells with interferon gamma, which is known to stimulate class 1 expression across all cell types. And you can see in our wild type cells, we have this nice shift of increased expression. But when we treat our knockouts, there is no shift behind background, suggesting that class 1 is completely gone. Class 2 is a little more complicated, as stem cells do not express class 2 at all, and class 2 is mostly expressed by antigen-presenting cells. And using the same color scheme as before, you can see when we differentiate our cells into a DC-like cell, our wild-type cells have an upregulation. Some it's only slight, but in activated cells, you get an even larger expression of class 2. But if you look at the knockouts, they look just the same as our isotype control, suggesting that they do not express class 2 as well. So far I've shown you that we've generated this HMKO knockout line, but the next question becomes, are these cells immunogenic? And this is a little early in the game for the question, but as it takes so long to make these edits and add all these proteins, we wanted to get our assays figured out before we moved on to the final line. And ideally, because these are human cells, we would be able to test them in a human system, but as everyone knows, you can't just go injecting cells into people. So we did do some experiments where we made humanized mice using cord blood. And in those experiments, even though we got decent human chimerism that's comparable to the literature, the mice wouldn't even reject our wild type cells, suggesting that they weren't as, immunocompetent as they should have been. So moving forward, we decided to use B6 mice. So B6 mice are immunocompetent mice. They have a fully intact immune system. And we do realize that by putting human cells into B6 mice, we're forcing them to cross a xenogeneic barrier. So going from a human into a mouse. But it's highly regarded in the field as a harder barrier to cross than an allogeneic barrier. So those are the reasons we stuck with using B6 mice and xenogeneic transplants, but I'm more than happy to talk about other reasons as well if people would like after the talk. So the assay we use for this is a teratoma assay. And how this works is we take our human embryonic stem cells, we inject them into the hind flank of a B6 mice, and we do this subcutaneously. Then we just wait. And over time, if our cells are accepted, the mice will form this little lump on their back called a teratoma. And because we're injecting stem cells, they can really differentiate into any cell types. We're not controlling by putting in an already differentiated cell type. So these mice just kind of form this weird glob on their back that is consisted of, different cell types, different tissues, whatever the environment, growth factors, et cetera, force these cells to differentiate into. Alternatively, the cells can be rejected, in which case the mouse does not form a teratoma on its back hind flank. So for the very first experiment, we took our H1 line, which is wild type, or our knockout line, so the HMKOs, and we just performed a teratoma assay with these cells. And what we expect is that the wild type cells will be rejected, as our B6 mice are fully immunocompetent. And we're not fully expecting our HMKO cells to be accepted, as NK cells should be triggered by the loss of class 1 and class 2. But we really wanted to know at this early stage, do we see any benefit of just knocking out HLA-1 and 2, and then MIC-A and MIC-B, although that wouldn't matter as much in a mouse system? And I'm going to take a little bit of time to explain how we show this data, as the rest of the data in this talk will be predominantly teratoma assays. But on these plots, the y-axis will be the diameter of the teratoma in millimeters. The x-axis will be the days post-transplant. So for this first assay, I'll be showing you up to 168 days, so over 5 months. The dashed line at 1 represents the limit of detection. So a mouse will be plotted at 1 if I can feel a teratoma, but I can't get a very accurate measurement that I'm confident in. If I can't feel a teratoma, they'll be plotted at 0. And for all of these experiments, they've all been done at least twice, and each experiment has 5 mice, so there are at least 10 mice done per experiment, or per plot that I show you. So to start off with our H1 cells in blue, you can see that, we put them in at day 0. By day 7, some of the mice have already rejected it, and all of the mice have rejected our H1 cells within 3 weeks. When we put in our HMKO, so the knockout line, you can see that our cells still get rejected, but now it's taking closer to 5 weeks for them to be rejected, and all of the mice still have a teratoma present in the first week. So far I've shown you that our wild type line is immunogenic in our B6 teratoma assays. If we put in our HMKO line, they become slightly less immunogenic. So what happens if we add our slew of inhibitory proteins? And for this, all of the proteins that we're adding are going to be mouse proteins because we're going forward with the mouse teratoma assay. So for phagocytosis, we've added CD47. CD47 acts as a don't eat me signal, and it binds to SERP alpha on phagocytes. Others have also shown that CD47 can inhibit NK cells, but that literature isn't as fleshed out as the phagocytosis literature, so I'm going to leave the CD47 by the phagocytes, but keep in mind that it could also be working to inhibit NK cells. For complement, we have CRRY, CD55, and CD59. CRY and CD55 work in the earlier stages of the complement cascade, and they prevent further activation of immune cells. Whereas CD59 inhibits at the later stage where a membrane attack complex is formed, and therefore CD59 prevents the formation of this complex, which causes cells to lyse. And for NK cells, we have H2K of B and QA1 as single chain trimers. And we have two versions of cells that express all of these proteins. One of them is called HMKO AAVS. This line, we have these proteins spread across two AAVS constructs, which target the integration of the constructs into the AAVS locus, which is considered a safe harbor and prevents silencing. Our other line is HMKO linty. In this line, every single protein is in a separate lentiviral construct. And that means that, we create the lentiviruses, we transduce the cells, but we don't actually know where our lentiviruses are landing and integrating. This can be a little problematic, which is why we prefer the AAVS line. But the benefit of the lenti line is that we can mix and match all of these inhibitory proteins, which will become more important later in the talk. But again, both lines express all of these inhibitory proteins. So as you can guess, we went ahead and we did a teratoma assay. Now with our wild type line, our knockout line, the lenti line, which is still on the knockout background, as well as the AAVS line, also on the knockout background. And here's just the data I've shown you before, and I'll animate in the next two, with containing all inhibitory proteins. So what we were shocked to find is that the lenti version and the AAVS version both grew up to about 2 millimeters in diameter, and they persisted for the entire duration of this assay. So again, 5+ months in the mouse. And honestly, when we got this data, it was before we'd seen any other promising data of, groups making lines that completely evaded the immune system. So we were incredibly shocked. And I don't think Deepta actually believed me. These mice we have done 50+ times, and they work every single time. So we were incredibly shocked by this result. The immune system is so complex that how were we able to outsmart it?
Yeah,
But
it's
anyhow,
very surprising.
yeah. But anyhow, you can see that our cells, the teratomas, plateau around 2 millimeters in diameter. So this led to the question, are we getting some immune suppression that is causing our cells to stop growing, or have they differentiated into a cell type that just can no longer proliferate? And I'm not
Hannah, just a quick question from Ben Schwartz. How many ESCs did you inject for your teratoma assays?
we do 1 million.
Okay, thank you.
And we do it in a matrigel membrane, so they localize to the area that we inject them in. So they shouldn't be spreading all over the mouse.
Right.
So to test if this was due to immune suppression, we had done another assay where we set up the same teratoma assay, but prior to injecting the stem cells and throughout the duration of the assay, we went ahead and we gave the mice depleting antibodies to deplete CD8 T cells, CD4 T cells, NK cells, and B cells. And we see the exact same results when we deplete all those cell types. So we don't believe that it is immune suppression that is causing them to plateau, but rather that they have just fully differentiated and can't continue proliferating at their differentiated stage. So an obvious next question is, are these teratomas actually the cells that we put in, or is there just some baseline inflammation that's causing this, what feels like a teratoma, even though it's just an inflammatory state? And so we took out these teratomas at day 102 after transplanting them. We sectioned them and we stained them. And on the top row, you can see our lenti line, and on the bottom, you can see the AAVS line. The first thing we stained for was human mito. So this will only stain human mitochondria. It will not stain mouse mitochondria. And you can see at both 20x imaging and 40x imaging that we get a nice human mitochondria stain, suggesting that these cells are actually human cells. We also had some conditions where we just tested some mouse tissue, and those do not stain with human mito as a verification. We also went on to stain for some of our mouse inhibitory proteins. So we did mouse CD47, CD55, and CD59. And again, you can see that all of these cells express these markers. And particularly for the MCD47 and the H-mito, they overlap, as these are the exact same sections being stained in these images. So great, we now know that the cells we put in are actually what we're reading out. So as a recap, we know that our HMKO line is slightly less immunogenic than the wild type line, but once we inhibit all of these other pathways, we've created a minimally immunogenic stem cell line. So, okay, we know that inhibiting all of these pathways is enough to prevent rejection, but do we actually need to inhibit all three of these, or are some more important than others? So again, we know inhibiting all three works, but what happens if we only inhibit individual pathways? So the names are going to start getting very chaotic, and I apologize for that, especially if you read the paper before this and tried to follow along. So hopefully we can get through this. So again, what we're using as our line that expresses everything is our HMKO AAVS line. Using our lentiviral constructs, we then generated an NK cell only line. We will be calling this line HMKO NK for our knockouts only expressing NK inhibitory factors. And it includes K of B and QA1, which were the original NK cell inhibitory proteins in our AVS construct. For phagocytosis, we're calling this line HMKO phago. And for this line, we have CD47, which was the initial marker we used in our AAVS constructs. But as we had kind of alluded to earlier, as more and more literature came out and other labs started doing similar experiments, we also added CD24, which inhibits phagocytosis by cyclic 10 expressing phagocytes, as well as PDL1. And I know PDL1 is most commonly thought of as inhibiting T cells, but it can also inhibit macrophages expressing PD1. So it still works as a phagocytosis inhibitory protein. And given our cells do not express class 1 and class 2, I wouldn't expect much effect on T cells, as the T cells are going to leave these cells alone anyhow. For complement, we ended up making 3 separate lines. The first line is HMKO comp, and this contains the original 3 inhibitory proteins we had in our AAVS constructs, so CRY, CD55, and CD59. We also made a line that only expresses CR1 or complement receptor 1. CR1 is thought to have an overlapping function as CRY and CD55, but not CD59. And then to make it up to a similar level as our HMKO comp line, we did a CR1 comp line, where now we have the CR1 replacing CRY and CD55, but we also have CD59 in addition to the CD59 up here, as CR1 and CD59 should not have overlapping function. And a lot of this came from the question of, could we actually minimize the number of inhibitory proteins we have on our cell? So if CR1 does work, it could theoretically replace CRY and CD55. So again, as you can guess, we went ahead and we did our teratoma assay. So we have our wild type H1s as a control, our knockout line, knockout with AAVS as our positive, this should definitely work. And then our complement only line is HMKO comp, NK only as HMKO NK, phagocytosis only as HMKO phago, our CR1 only, which is another complement line, HMKO CR1, and then our final complement line, HMKO CR1 comp. So we're going to go through this data slowly, so hopefully I don't lose people in the chaos of naming.
It's good. You're doing a great job so far guiding us through.
It was really hard to write for the paper, good luck. So now I'm going to start showing you this data additionally as, survival plot type of plots. And this is because as you will see, there are a lot of lines that start adding up, and then it could be easier to look at as when do mice drop out. So the growth plot is going to be the same one we've seen before, but now what I'm showing you as the persistence plot, the y-axis is just going to be the frequency of mice that still have a detectable teratoma. So as you can see, they all start off at 100%, and then our H1, so the wild type line, drops off pretty rapidly. The knockouts are delayed, and then our AAVS cells, we never drop off. Okay, so going through one by one. So our HMKO comp line, so complement only, is now in pink. And you can see that these teratomas do actually grow a little bit, whereas our knockout cells don't. But they're still rejected pretty rapidly. It's delayed relative to the knockout line, but again, they're not surviving like the AAVS cells are. So this suggests that our complement inhibitory proteins are giving the knockout cells a little bit of a boost, but it's not actually enough to bring them to the level of our AAVS cells. If we then animate in the NK only line, so HMKO NK in purple, you can see that this pretty well overlays the comp line. So this also suggests that the NK cells provide a little bit of a benefit, but again, it is not enough to get these cells to persist. When we move on to the phagocytosis only line in orange, you can see this is how it's getting hard to see with all the lines. It pretty well overlays our knockout only in red. And you can see this over here as well, maybe a little more clearly since it's not a complete overlay. And the one thing I'd like to point out with this line is there is another group that has generated a line that is class 1 knockout, class 2 knockout, and contains only CD47, and they have had success with their line. We don't see it in our data, but I think there's a lot of variability that's dependent on, the cell type used, as that group uses iPSCs and we're using embryonic stem cells. Additionally, that group has gone on to do some differentiations, and we're still just doing this in stem cells. It can also be due to the site of transplantation, as there are going to be different immune barriers at different locations during transplant. And it can be due to the differences in models, as all we've done right now is the B6 mice. But then moving on to our knockout CR1 line, which only expresses CR1. Again, this doesn't provide a benefit over the knockout line alone. You see it overlaps again with just the knockouts. So CR1 alone does not provide any benefit. However, if we do CR1 with CD59 in our CR1 comp line, it's beginning to look the same as our knockout comp line. So again, there's some benefit, but it's not enough for overall persistence. And the fact that this mirrors our comp line suggests that CR1 can replace CRRY and CD55. And additionally, CD59 is required, given our CR1 line did not provide any sort of benefit. So moving forward, what we found from this experiment is that our phagocytosis only line did not provide any benefit relative to our knockout cells. Neither did our knockout cells expressing just CR1. However, if they express our whole slew of complement inhibitory proteins, or CR1 in addition to CD59, they do better than knockout only. Our NK only line also does better than knockout only. So because these two large groups of inhibitory proteins did provide a benefit relative to our knockout line, we wanted to know, can we make a line that only expresses NK inhibitory proteins and complement inhibitory proteins, and does that work as well as our whole slew of proteins in our AAVS constructs? And so as you can imagine, we used our lentiviruses to generate two more lines to ask this question. So the first one will be called HMKO, NK, and comp. And this is the original complement inhibitory proteins and NK inhibitory proteins found in our AAVS constructs. And then as you can guess, our other one is the CR1 version. So it's still NK and complement, but we have CR1 replacing CRRY and CD55. It still has CD59, K of V, and QA1. And again, as you can probably guess by now, we threw this into a teratoma assay, again in B6 mice. And so now what you can see is when we animate in our NK and complement only line, it mirrors AAVS, both in growth and persistence. The same holds true with when we do our CR1 NK and complement line, that it grows similarly to AAVS and it persists just as long as AAVS. So again, this recapitulates that CR1 can replace CRRY and CD55. And it also shows that you need to inhibit both NK and complement. And I'm not going to show you the data for this today because it turns into a pretty big headache pretty quickly. But we've done some experiments where we have C3 deficient mice. So those mice are deficient in a key aspect of the complement pathway. And we've done a mix and match of that with NK depletions and a mix and match of, our knockout only, our knockout NK inhibitory line, and our knockout complement inhibitory line. And we can mirror those results in those mice. So it provides another layer of evidence that you need to inhibit both NK cells and complement to get these cells to persist. And so in summary, we knew before this that we had our minimally immunogenic line if we inhibited phagocytes, complement, and NK cells, in addition to the inhibition of T cells from the initial class 1 and class 2 knockout. And we've added to this by showing that you really just need to inhibit NK cells and complement. Inhibiting phagocytes seems to be pretty dispensable. So obviously, we do not care what happens to stem cells when we put them into mice. So can we differentiate them into a actually useful cell type to put into a B6 mouse that would be more, mimicking what you would expect to do in a person? And I will say that, science cannot be done in a vacuum, and our lab is very immunology driven. So we can differentiate these cells into immune cells, but that's not the most useful cell type to try to put into a mouse and find later. So we turned to one of our collaborators, and I will be upfront, this is orphan data, but it could be very interesting to this crowd. So our collaborator differentiated our human embryonic stem cells into beta cells. And he did this for both our knockout line and our knockout AAVS line. And he transplanted them under the mammary fat pad of B6 mice. He also put in a GFP tag so that he could easily find these cells when he pulls them out. So I'll show you sections of the area that he transplanted these cells. So for the HMKO line, you can see that there is DAPI staining, so he is actually staining tissue sections, but there's no GFP staining and there's no insulin being produced. And this is at 1 week. And in the AAVS line, you can see that our cells are still there, given you can see a nice GFP signal, and they are producing insulin. And what was even more exciting, because 1 week, how well does that really translate? He also looked 8 weeks later in the B6 mice, and this is the AAVS line, and you can still see the graph that he had initially put in, suggesting that these cells are able to persist as beta cells in B6 mice. So just as a final summary, we created our minimally immunogenic human embryonic stem cell line by knocking out class 1 and class 2, MIC-A and MIC-B, and adding a slew of inhibitory proteins. Of these inhibitory proteins, the proteins we added for phagocytosis were not very useful, and we could get by with just inhibiting NK cells and complement. And when our collaborator differentiated our cells into beta cells, those also persisted in B6 mice. So looking at future directions, there are obviously a ton of options for where to go next. But one that's of particular interest to us is, can the modified stem cell derived beta cells persist in and treat NOD mice? So if we add that added barrier of autoimmunity, can they still persist? And I'm not going to show you the data because it's very preliminary and it's our collaborators' data, so I don't want to steal their thunder. But they did put our cells into NOD mice. They waited 3 months and they were able to find them after the fact, and they were still producing insulin. So it does suggest that, these cells can actually work in an autoimmune setting. Additionally, we want to be as transparent as possible and let this process work for anyone that has any other cell type of interest. But that also has us wondering, can we get these edits to work in other cell types, tissues, organs, and can it work to treat their disease models of interest? And because of that, that's why we're trying to be as transparent as possible and getting all of our resources out for other people to try. And additionally, this is more of a long shot question. How well does this work in humans? And I just wanted to point out that we have made the human orthologs of all of our mouse proteins. Some of them do not have direct orthologs, especially the NK cell ones, but we've made what we believe to be the best equivalent possible. And I can say the human proteins, we've tested some of them in vitro and they appear to at least work. I mentioned that our humanized mice didn't do that well in rejecting even wild type cells, but I think as mouse models get better, maybe there's a better opportunity in the future. Additionally, this could translate into non-human primates, but a lot of this just depends on what the future holds and how the field develops. And with that, I just wanted to provide some information on our paper. So I've hit the highlights today, but there's a lot of stuff that we couldn't include. And we're trying to put our paper out there, as a roadmap for anyone else. If you don't want to use cells on an H1 background, that's fine, but now you at least know how we made our edits. And even if you don't want the same edits as us, you can go through and make your own. We also hit a lot of roadblocks in doing this. There's a reason it took 10 years to do. So we learned a lot about along the way, and we included those roadblocks in the paper for others to know. So for example, in 2016 and 2017, a few papers came out saying that when you use CRISPR-Cas9 to edit stem cells, you end up getting a lot of stem cells with p53 mutations. So we learned the hard way and we went back and sequenced after that. And early on in making our edits, we picked up a p53 mutation. So all of the work we had done up till then, it was still valuable information, but we had to go back and remake all of our lines and redo all of the experiments. So we want
That's
that.
why it's a great thing for negative data to be published, which it hardly ever is.
Yeah. So
Oh.
this was a very long process for us. So if we can save someone else time, we hope we can.
That's great.
Additionally, we're making some of our lines available through YCell. We're still in the process of getting these out there with all of the, legal paperwork and all of that. But the lines that we're planning on making available would be the HMKO line, so the line that doesn't express class 1 and class 2, as well as our edited line for the knockouts that expresses all of the mouse proteins via the AVS construct or the lenti constructs. And then we also made the line that expresses all the human AVS proteins as well. So hopefully in a few months, those will be available and people can buy them as they need them if they would like them. Additionally, all of the plasmids that we made in this paper, we have put on Adgene, and you can find them by either searching for Deepta's name. I understand it's a very complicated last name. Or if you look at the paper, we have listed the formal names and the IDs in the paper, so they're easier to find. And the plasmids that we have made available are our mouse and human AAVS lines, which are shown down here, as well as our lenti plasmids, again, both mouse and human. And those ones, I don't know if you can see the things popping up, are only for one gene at a time. So even if you don't want to make this whole slew of edits, you can go through and say, hey, I just want to add CD59 to my cells, and that construct's available. So we really we want our learning experience and the resources we created to be a toolbox for others. So take that as you will. And, even if you don't want these proteins for inhibitory or for making like a minimally immunogenic cell line, you just care about CD59, CD55, proteins are there if you want them. And I'll bring the slide back at the end once I get through acknowledgements if anyone wants to look it over again. But with that, I'd like to thank a ton of people as, science isn't done in a vacuum and we're all really limited by our own skill set and the expertise of others is always wanted. So I'd like to thank, Deepta especially. He had this idea before I even joined the lab and, the edits were already starting for making our knockouts by the time I joined. Additionally, other lab members that have heard me talk about this ad nauseum. We have great collaborators at the New York Stem Cell Foundation. So Paula and James actually did our IHC for us in the teratomas, and they're carrying on with some beta cell work. Bjarki did the data that I showed you at the end of just the beta cells in B6 mice. We wouldn't even have these lines if we didn't have considerable help from the Genome Engineering and IPS Center at WashU, and some of those members went on to move to St. Jude after the fact, but they continued helping us out. So they helped generate our knockout cell lines for us, and then they also helped fix the P53 mutation, which was great. Additionally, our complement advisors were the Atkinson lab back at WashU, and our NK cell advisors were the Feniger lab also at WashU. And science would be nowhere without our funders. So we'd like to thank JDRF, New York Stem Cell Foundation, Bill and Melinda Gates, as well as NIAID for their help. And with that, I will take any questions.
Fantastic. If anyone has a question, we've got something from George Harb. Hannah, beautiful work. Congrats. Have you or your collaborator compared your lines to the Pluristics Pancella Nagy lines on the effectiveness of immune evasion?
Can you repeat what the line was again?
Yes, the lines to the, it's right in the chat too. If you look down, the Pluristics backslash Pancella backslash Nagy lines.
Yeah, we haven't gotten those lines, but I know the lines from the Nagy lab. We had done some edits to try to get similar to theirs. And some of them we do see similar levels of immune evasion, but not all of them. I can say that ours aren't an exact mirror of what they did, but they're as close as possible. And the Nagy lab also had found too that, the level of expression mattered a lot more. So in one of their papers, they made all these edits and then they pulled out the cells that worked and found that the varying levels of expression played a role. So some you had to hit a threshold to make it work. And we haven't gone down that route of seeing, do we actually need to hit that threshold? We do know that in our lenti lines, we tend to get higher expression than our AAVS lines, but we don't see any difference in the growth of those lines. So I don't know how much that necessarily matters for our constructs. But yeah, we don't have a direct one-to-one comparison of our cells with theirs.
So thank you. Yeah, and I mean, so what you're heading out basically out the door, hoping to, get a position as you stated earlier in industry. Who's going to take up the project in the laboratory once you're going?
So we kind of view this project as, I don't want to say complete because there are obviously a lot of other directions this can go, but a lot of those directions we feel like are out of our hands.
Hmm.
So, our lab, it's mostly a B cell lab.
Yeah.
So our lab is working on differentiating stem cells into B cells, then long-lived plasma cells, with the idea that if we do this, we could make a long-lived plasma cell vaccine and make it specific to, say HIV or flu. Um, but in terms of the immune evasion itself, I think we're happy to serve as advisors to others. But that's part of the reason we're also just trying to get our cells out there. So, if people want them to try them in their system, it's completely open.
Yeah,
You
as
know,
a resource. That's
yeah.
really admirable.
And if, they say they don't want them on the H1 background, they now have our, protocol effectively on how to make it in the line that they want. If they don't want the entire mix and match of all these plasmids, they can do their mix and match with the lentiviruses we produced or the constructs that we made. Or it can also just serve as, hey, they want to make a different inhibitory protein, but we've at least given some sort of framework.
Yeah, no,
So
it's
we're happy to advise, but experimentally on our end, I don't think there's much more we have planned for, the stem cell immune evasion itself.
I see a bunch of people in the audience from industry. I wonder if anyone else would like to unmute and ask a question, or maybe you can have
Hi, I can ask a question. Hey
great.
Hannah, this was a brilliant talk. I'd read your paper, but just having you explain it was amazing. So
Yeah,
a
it's a
question.
lot.
Yeah, I read it over the December break, so it was a great reading.
And just so
Um,
people know, this is Surby Jane.
Yeah, hi everyone. So my question is around the edits that you've in place to overexpress. You have CD47 there and other targets. Do you have a sense of what thresholds are required to surpass the barrier? Or is there, do you have a sense of that?
So I can't give you an exact, like, this many molecules of CD47 have to be expressed, but I do know that when we do our staining, this is kind of going to be a little vague without the flow plots. But in our AAVS construct, say the staining for our knockout line is below 10 to the third MFI. Staining for our AAVS line will be between like 10 to the third and like 10 to the fourth, maybe fifth. And that's enough to get evasion. Our linty line, it's closer to, 10 to the fifth, maybe a little higher. And that, I don't think you're going to reach a point where you have too much expressed. But we at least know having that, full shift over background is enough. So not the best answer since we haven't actually like quantified how much is there, but I think there needs to be at least some level of threshold.
That's a nice experiment for someone to do next.
Yeah, thank you.
The lentivirus is on Adgene if you want it.
I have one more question, and that's around, your comment on P53 mutations coming with CRISPR. That's definitely true. The other literature out there is on TP53 coming in ESCs, in fact, with just culture without any editing. So, despite that one sad, outcome of you getting TP53, but then fixing it, that was brilliant too. Are there other instances? Have you checked for genomic integrity of these lines over time? And did you see any other or those mutations pop up again?
So we haven't looked, extensively over time in these individual lines. We have made some other lines in the lab that we're not seeing P53 mutations pop up in. And then with these lines specifically, when we did the sequencing, we looked at everything. And, obviously there's going to be an occasional mismatch somewhere, but the only concerning mismatch was the P53 mutation that we found.
Awesome. Thank you.
Yes.
Great. Here's another question. Really extensive work. Congratulations. Oh, this is Scope Nair from the Bay Area. How do you reconcile with allo versus xeno context in your edit selection? Perhaps for the allo context, you may not need all of those edits you found in the B6 background.
Yeah, so we do realize that that's going to be different between mouse and human. Um, the problem, as I had mentioned, is we tried to make these cord blood humanized mice. And even though we had pretty robust T cell, B cell chimerism, we also had myeloid cells, they just couldn't reject. So there are different humanized mouse models, and some of those, they can make one aspect of the immune system better, but not all aspects. So there are some that are really good for NK cells, but they're not necessarily great for T cells. So I think in a lot of the humanized models that exist, yeah, that's fine if you want to try it to test it on NK cells, but it's hard to see the whole picture of the entire immune system. Another option that people have been going to is instead of doing cord blood humanized mice, they will take PBMCs from people and put those into immunodeficient mice.
Oh, that's the next question. Did you try PBMC humanization?
So where that gets really messy is you start getting graft versus host disease. So we haven't actually done it ourselves, but in a lot of papers that do it, they'll put in the human cells. They're fully matured, so they don't have to go through what the cord blood humanized mice do of trying to develop human cells in a mouse environment. But then you get the human cells attacking mouse tissues. And in a lot of papers that do this, the mice will actually die in about 2 to 3 months, or they'll reach points that you have to terminate the study. So I think it could be a useful model for short-term studies, but I don't necessarily think it's the best model for long-term. But you could still at least, put your cells in and see if they make it to that time point where the mice start getting sick. So that is one option, but we have not necessarily explored that.
And the comment is that leaves you with BLT, BLTS models, correct?
Yes.
Mhm. One more. PBMC have shown robust skin allograft rejection.
Okay.
Commentary.
Good to know.
Yes. You really simplified a very complex series of experiments and a long trajectory of work. And we're really we're able to act as an excellent guide through this paper. So thanks so much for joining us and for, fielding all these questions and wishing you the best of luck in your next venture. We're going to be keeping an eye on you. It's a very meticulous study. So
Thank
thanks
you.
again for sharing.
Thanks everyone for coming and thanks for the invitation, Monica.
Okay, bye-bye now. Have a great rest of your day.
Thanks, you too.